Patricia, Izar and Nicolas Chaline will be presenting their research about comparative study of development and environmental effects. So thank you very much for coming, and it's your turn.
Thank you. Bom dia a todos e todas. Good morning. And so Yarka has already introduced it, so I have to get used with this. Let's see. Yeah. Well, so just to you understand what we are talking about here. So we are focusing on the developmental question and considering the environmental contributions and structure that channels the emergence of patterns of behavior. So we are proposing a dialogue considering some species, basically capuchin monkeys and ants. So I'm going to talk a little bit about the ecological approach, and I will present something about capuchin monkeys and learning the cracking behavior. Patricia will also talk about capuchins from Piauí state in the northeast of São Paulo, and Nicolas will talk about ants. Okay. So when you think about behavior and learning, we think about the contexts of environment, and thinking about humans, for example, we always think about tool use and objects and environment and things that channel behavior and actions and learning. Here I'm using the perception action perspective. You can see Professor Lockman in the picture. And he has a paper where he says that tool use is not something that pop-ups in the maturation of an organism of a human being. He was talking about humans. But it is a continuous phenomenon along development, and it's rooted on the perceptual learning. So subjects explores, manipulates objects, and then tool use is the result of learning the affordances of the objects and the tests. But we are focusing on capuchin monkeys learning how to use tools. Specifically, you can see a picture here of a capuchin monkey from Tietê Ecological Park. The capuchin monkey is cracking a Ciagrus nut using a stone which we call hammer, and the stone where the monkey places the nut we call anvil. And we have the social context also, and we are talking a little bit about it and how it influences learning. Something more about perception action perspective. I'm quoting here Esther Thielen, an important researcher. She was psychology, and she studied motor development in human infants. And she says that movement is not determined by the nervous system alone, but it emerges out of the confluence of circumstances. So movement is not in the infant in terms that it's not an instinct or something that is a fixed motor program or reflexes, but it's contingent on the environment. So we are using this idea to learn something about capuchin monkeys' development of motor behavior. Not only motor behavior, but how something more about nut cracking and the emergence of nut cracking behavior. This monkey here is from Fazenda Boa Vista in the northeast of Brazil. You can see that we washed the monkey is also going to crack a nut there. And but the nut is different from the other that I showed you before, and the hammer is always is different also. So these different objects, they impact in the way the learning acquisition will happen. And we will also use the idea of niche construction, and we have Leila and Adeline Smith that work with it, but I like Richard Lewontin sentence that was, well, he said, living organisms are not passive entities. They actively build the environment and so the selective regime in which they live and reproduce. Meaning that when a monkey, we are thinking about monkey, let me return to the monkey. When the monkey cracks the nut, it leaves rests of the cracked nut and leaves the stones and leave marks and the anvil substrate that may be a stone or a log. In this example here, it is a log. And we also have the social contest of cracking nuts. So when somebody starts cracking, the others usually are around. Some start cracking also, and so they are curious about it, and so nut cracking may emerge or may not. So they crack nuts. Here you see the example from Tietê Ecological Park, and here, how do we use it? Let's see. I don't know. Let it be. So at Piauí. Some pictures showing you some details about the hand of the monkey, the nut, the stone, the size of the stone, the movement the monkeys have to do to crack the nut, and the social contest. You see a little one watching the proficient monkey cracking, and you see the little one here on this other picture exploring the cracking site. And here you have more examples. This picture, you can see the movement. You can see the tail, that it is used to give support, and how the monkey has to lift and then go down, and the knees, they have to squat. I don't know if it's the correct verb. And so, learning to use to use is not a simple thing, it's a complex behavior. And in social life, they have opportunities for learning. So this, you can see the Brazilian map over there, and the Piauí state in red. And then here you can see Gilboa City in the south of Piauí state. That's where we study these Capuchin monks that I'm talking about. And in the picture you can see a nut cracking site. I think it's important to notice that you have there some stones that were left by previous crackers there, and you have some concavities and holes in the anvil stone which guide how to place the nut. And so we think social learning is something broad. It's not. It's wider than only watching somebody cracking, but we have all this niche that was constructed and impacts the emergence of nut cracking behavior. These are some examples of the nuts they crack there. And we know that the reuse of tools that were previously is used for proficient monkeys, and the scrounging of the rests of nuts that were left there, they attract the other monkeys to the sites. And they are, so we think they are important elements to understand the emergence of nut cracking. In a previous paper, we it's called the fourth dimension of tool use. We are thinking about the importance of leaving these artifacts, these objects, in the nut cracking sites, and we gave some examples of how it could aid the learning of tool use in capuchin monkeys and also in chimpanzees. So here this is a picture that I've already shown, but you can see the hand over there, the rests of the cascas. I forgot in English. You can see what is dead there. So they were left there, so young monkeys are attracted to these cracking sites. And where nut cracking is a tradition, and now we are talking about animal traditions, we see that they use the sites, they use what was on the sites to explore, and I wanted to highlight how they change the environment, and it canalizes the behavior of the next monkeys who will explore what is on this site. So more beautiful pictures, because we love monkeys and we think they are cute. We cannot decide which picture to use. And so, in another paper, we found that the variability in actions, considering actions that are important in nut cracking sequence of behavior, we see that variability declines as the monkeys get older, and also as the monkeys get proficient in nut cracking. So it means that when they learn how to crack nuts, they focus their behavior on the more effective sequence that will lead to opening the nut. But now we wanted to study how do the use of stones for cracking and scrounging behavior developed. This is a juju monkey. I have to give the credit. One student of mine, Flavio, he created juju monkey, so this is our pet from the lab. So we studied how objects influence the development of tool using skills in a wild population of brown capuchin monkeys from Piauí. And this is how it looks like. And we wanted to investigate the reuse of artifacts and the scrounging of nuts left by the individuals on nut cracking sites. Because we already knew it was important, but we didn't have a systematic study about it. Or we had, but not, we wanted to, we had more questions to answer. So this is published in Evolutionary Human Sciences. It is in collaboration with Andrés Ballesteros. It was his PhD. And with Patricia and Elisabetta Wieselberg and Dorothy Fragazzi. It's part from Eusebio's project, maintained by Patricia. And so we went to the field in 4 different field seasons, and we stayed in a place where monkeys used to crack nuts spontaneously. we used 7 cameras that were disposed, as you can see in this draw, and 33 capuchin monkeys arrived, and we filmed these monkeys spontaneously using the stones to crack the nuts and to explore also, and we used Boris software for transcription. 3 minutes, it means I must end in 3 minutes, okay. Thank you very much. So this is how it looks like. And we registered the frequency of stuns and nut manipulation and scrounging, if they were used before by another monkey, if they were reused immediately or 30 seconds after the previous monkey left, and we registered if the monkey cracked the nut or not. And we, considering that the objects left on the side attracts the monkey's attention, considering the variability of actions decreases of proficiency, we have the following hypothesis. The monkey's perceptions of nut as food item and stones as tools change throughout development as a function of the acquisition of proficiency in cracking. It means that when a monkey initially arrived on a cracking site and did not know that he could crack a nut, that stone over there would only be something from the environment. But after learning how to crack nuts, that stone becomes an artifact, becomes a tool. And so I'm going faster through these, our variables, dependent variables, and our independent variables. So we register the frequencies of reuse, scrounging, when the monkey was cracking, when the other monkey had already left, and we had the age of the monkey, an index of behavior variability, and the level of proficiency of the monkey, if he was naive or not, proficient or not. We use generalized linear models to analyze, and our results were the following. So considering proficiency, behavior variability, and age, we found that the older, the more proficient, the younger, the more behavior variability the monkey had. And it was a confirmation of a previous study with a different population. But behavior variability was a better predictor of proficiency than age, meaning that what was more important here was not the age itself, but how the monkey was focusing on the task of cracking the nut. And we also found about delayed scrounging that the behavior variability was the best regressor, so it was positively correlated. So when the monkey was not focusing on the sequence of behavior necessary for the task, he used to scrounge more when other monkeys were not around. And about the immediate reuse of hammer and anvil, we saw that as individuals grew older, they decreased the immediate reuse of hammer stones. It means that, well, we can talk more about it later, and if you want, you can make questions, and then I can explain you in more detail. And about delayed reuse of hammer and anvil, we saw that objects used as artifacts in the absence of others. So, because we saw that the monkeys were reusing the hammer when they were more focused on the sequence of nut cracking, the best sequence of behavior used for cracking. So in conclusion, behavior variability was the best predictor, considering our hypothesis. Monkeys were increasingly likely to reuse tools as they were increasingly focused on cracking behavior. And we think that objects become artifacts or tools as nut-cracking behavior develops. And the social context establishes the elements of the cognitive niche marked in the physical world. And the process of cognition results from the interactional dynamics involving nuts, stones, and each animal, what each animal can do, and considering its ages and experiences. This is just, I just wanted to show you, if you want to learn more about capuchin monkeys from Fazenda Boa Vista, you can find it on YouTube. And that's it. Thank you very much. Thank you for people who helped in the field, for everybody who funded us, and well, that's it. And that's it. So, wow. I think we can move to Patricia, and then Nicolas, and then we sit over there, and then we have all the questions. Okay.
Good morning, everyone. Bom dia para todo mundo. Thank you for being here for our symposium on development. And today, I'll talk to you, I will briefly summarize my main research on social systems of capuchin monkeys, and then I will talk a little bit about a set of studies on the development of these social systems or their social behavior. So, I don't know if everyone is familiar with the robust capuchin monkeys. Of course, Bri has just spoken about the capuchins in Piauí. But in fact, capuchin monkeys or the robust capuchin monkeys refer to nowadays to 8 species of the genus Sapajos, and the entire genus occur in all biomes of South America, and they occupy areas with very different ecological features, and they present correspondent variability in their behavior. There has been a lot of studies investigating their plasticity, a lot in their, oh my God, in their modes of locomotion, and especially in their feeding behavior. But in our group, my study group, we have been interested in the variation of their social systems in relation to the ecological features. And we have shown that features of social organization, mating systems, and social structure reflect differences mainly in the risk of predation and the features of food sources they use. In particular, group size and sex ratio vary in relation to predation risk. The dominance hierarchy, dominance relations among females vary in relation to the features of food sources, and the dominance relation between the sexes, between males and females, reflect differences in demography, in sex ratio especially. But today, I will focus on the comparison of 2 populations that me and my group, we have been studying for more than 15 years now, 12 years in Bahia and in Piauí. Briseida just spoke about the population of Fazenda Boa Vista that we study in Gilboés. This is the northeast region of Brazil. It's an area of the ecotone between Cerrado and Caatinga. So, as you have seen in the pictures that Bri showed, it's a kind of dry environment. and the population of Sapajos xanthosternos that occurs in the south of the Bahia state. So it's northeastern region, you said northeastern of São Paulo, but today I will talk about these two populations of the northeastern of Brazil. One that occupies an area of Atlantic forest, and one in the more xeric environments of Cerrado and Caatinga. So I will summarize our main findings across all these years. So we have shown that group size in Una is much larger than in Boa Vista, and this reflects the fact that there are more females in the groups, more females associating groups in Una than in Boa Vista, but we also have proportionally more males in Una than in Boa Vista. So we have higher group size and sex ratio in Una, the area of Atlantic forest, than in Boa Vista. And this happens in spite of similar conditions of food availability in both areas. In fact, here we have food availability in Boa Vista and overall similar to Una, especially due to patches of plantations that occur within the Atlantic forest, plantations of cocoa and jackfruit, and then the oil, and the monkeys feed a lot in these trees. So in spite of similar conditions of food availability, we have differences in group size and sex ratio. This, in the literature, usually is related, especially the higher sex ratio, to a higher perceived risk of predation. What is exactly what we see when we compare both populations based on the frequency of alarm calls that we record that the monkeys emit. So monkeys in Una, they perceive, based on their alarm calls, they perceive more or less twice the predation risk than in Boa Vista. And this is because of the presence of the arpia, which is a super large bird that predates the capuchins, but also due to still hunting by humans. They give alarm calls for signs of hunters, such as dog barks. We have also shown that, esqueci de marcar meu tempo, né? Eu ia marcar, tá indo tudo bem? We have also shown that the dominance relations among females in Boa Vista are nepotistic, are linear, nepotistic, very stable across years. And in Una, we almost cannot see dominance relations, just for a few. The dominance hierarchy is almost absent in this area. Again, in spite of similar features of food sources. However, we can see here that in Una, more members can feed at the same time in the same tree, probably when they are using those jackfruit and the oil trees that I told you about. Regarding grooming relations, in both areas, in spite of the dominance hierarchy in Boa Vista, we don't see any relation between dominance and grooming relations. Relations are reciprocal, equally reciprocal in both areas. The main difference is that in Boa Vista we see a slight modularity, while in Una everyone grooms everyone, so it's a super dense grooming network. And finally, we have shown recently that the index of female dominance over males in capuchins varies according to sex ratio. The higher the sex ratio, the more dominant females are in relation to males. However, you can see here in purple that in Una, this female dominance is below the expected by this relation. So overall, when we summarize this data, we can say that in the population of Una is kind of more egalitarian, relaxed, and more affiliative than the population in Boa Vista. We then turned our attention to understand whether these differences are a consequence of an innate temperament, if these species are different because some kind of genetically inherited difference, or if we could identify differences in the developmental trajectories of these populations within these different contexts, social contexts where they are growing. For that, we started to, in 2013, almost 10 years already, we started to film every newborn in each of these populations. We tried to have one day, focal filming day per week from the first week until they complete, they were 3 years old. So 36 months, filming 1 day per week, every week. Of course, we have a lot of data missing, but this is what we tried to do. And we have babies here in both populations. So I'll first show the work of Irene Derval that is sitting there. She was my PhD student, and I'm reporting here her incredible work. Investigating the personality traits of the newborns or the youngster, the initial development of 12 individuals from Una, the Cepajos xanthosternos, the egalitarian population. She investigated 9 males and 3 females, and from all that video database, she selected 10 developmental points, the first 3 months, then the 6 years, the 9th, the end of the first year, the end of the infancy, and then 3 points in the 2nd and 3rd years of life. the time goes very fast. So she had almost 5,000 minutes, and then from the 44 behaviors she selected the most frequent 15, and with that she conducted a principal component analysis, and she found 4 behavioral axes that we called sociability, that is mainly related, several features here, but it's very importantly related to the time spent with neighbors around, anxiety that is relating to eating and scratching, openness that is related to play behavior and sexual behavior. In young, she was studying individuals until the 3rd year of age, and activity that is related to moving, walking. We can see here that individuals varied a lot across the developmental points in these 4 traits. She found overall certain significant repeatability in sociability and anxiety. It's too small, I don't know if you can see. But what is important, she found this significant repeatability, but values were below 0.4, so it's kind of not so repeatable. Repeatability is a measure of the rate between intra-individual variance and inter-individual variance. So when we say repeatable, it means that the individual is consistent and there are consistent inter-individual differences. And we can see here it's showing what is the months where this repeatability is significant. So we can see that in sociability, we have repeatability in the 1st year of life, and then it disappears to reappear in the 3rd year. For anxiety, we also have this variability in the first 3 months. This is not appearing yet. Nothing almost for openness, and anxiety also varying across the developmental points. So they are very unstable in this beginning of their life. So we say that personality is not established yet in the first 3 months 3 years of their lives. The same, more or less the same result. Oh, I forgot to, the first 3 years, I forgot to correct this one. So Julia Omena did her master's following more or less what Irene did with the population of Una. She studied the monkeys in Fazenda Boa Vista, but looking only to the sociability X of personality. And she was looking to the initiation of social interactions, social behaviors by the immatures to try to investigate whether this social personality would be established. She repeated the procedures that Irene developed for the study in Una. She did not also find repeatability. Only she found only repeatability, significant repeatability in the 2nd month of life and the 9th month of life. So again, this we cannot say that they have a personality, consistent social behavior yet in their first 3 years of life. Then these 2 other studies, 2 master studies, Marie Caroline, Franco Rogério, and Emily Faverin, they studied the development of the association network in these 2 populations. They used the same video database, and they recorded the neighbors within 1 meter of the individuals, and they developed the social networks and compared the development of these these network metrics, degree, strength, density, and modularity. And Marie also found this great variability across development. And so that we could only see a significant difference in strength between the first and 2nd month of the individuals and the other developmental points. She found no significant difference in degree, density, and modularity across the first 3 years of life. And this difference in strength disappears when remove the mother from the network. So we saw a stability in social development across the first 3 years in Boa Vista. Almost there. I know I'm late. The same thing Emily did with the population of Sapajos xanthosternos, the same measures. We see even more variation here. But here, Emily had to reduce the developmental points. She had more developmental points than the individual, so we conducted a cluster analysis, and we compared the development across these clusters of development. So we have 3 points for strength and 4 for degree and density. And again, we find a significant difference from the 3 first months of life and on. So we don't see a great difference in the development, the social style of both populations, but development seems to be faster in Una in comparison to Boa Vista if we consider this difference in the first 3 months of Una in comparison to the rest of the first 3 years, while in Boa Vista they are always more or less the same. And we attribute this difference in the rate of development, in the pace of development, Flavio, we don't attribute this to inherited differences, but to these, again, to the difference in predation risk. It's common in primates, even in human primates, that in conditions of higher perceived risk for the babies, mothers promote independency, earlier independency than in areas with lower risk. So sorry for going over my time, and that's it. Thank you very much. Thank you, all the students and field collaborators, the funding agencies. Thank you for being here.
All right. Good morning to all. Thanks for coming to this event. Thanks for the organizer for inviting us to present some of our research, which is not about human animals. So when Briseida proposed that we did this symposium, I was thinking, what could I talk about? And I thought that some work I did some years ago about how development is influenced by the rest of the group would be something interesting, because there is some concept associated to that which actually can be applied to any living organism, more or less. So I will present my work on the clonal ant, Oceraya biroi. The idea behind this research actually is, although development has become a fundamental study area to understand behavior, and it must be obvious to who works on humans, psychologists, it's less obvious perhaps when you think about invertebrates such as ants, insects, and even microbes. What happens is that people working with humans actually sometimes they get a bit set back by some ethical problem, by the facility to use this organism in controlled experiments, which in ants we can actually set up pretty much whatever experiment we want. So it's something which that we take advantage of to do research on some more complex mechanism associated with development and social behavior. And last, before I start presenting the work on ants, I think something we may not always remember is that our environment, and perhaps the most important factor in the environment that will influence us, is other organisms from the same species. So in humans, we interact every day with lots of people which are not related, which are work colleagues, and surely this is something that will influence our future behavior during infancy as well. This surely will influence what adult we will turn. And this is something that actually, in my opinion, is true of whatever organisms that interact in groups, that the proper interaction creates an environment which actually also modifies the way the environment is perceived by the organism through the filter of the social group. So I think this is one of the important messages during my talk. So before I start, I will just present three of my collaborators in this project. My two PhD students, Serafino, Emmanuel, and Daniel Kronauer, which is now in the Rockefeller Institute, University in New York. And he's continuing work on the genomics of social behavior using this same clonal radar ant. So if you're interested in this type of work, look at the website of Daniel. He's publishing loads of stuff on genomics in ants. So at the basis of my research actually is cooperation and conflicts. So you all know that Maynard Smith and Satsmari, when they work about the major transitions in evolution, they saw that the complexification of organisms from genes to cells, cells to multicellular organisms, and multicellular organisms to societies, which actually have division of labor, which have a difference between reproductive individuals and not. It's something that is important to the success of organism, but it's something actually we have to deal with as biologists, because also it seems to bring some advantages. There's also a lot of existing conflict that comes with the fact that stuff is living together, be it genes, cells, or organisms. So these conflicts, which are one of the mainly studied problems in social behavior and social evolution, is the fact that societies are heterogeneous, heterogeneous, and the interests of individuals inside the societies can be different. Actually, between societies as well, you can have conflicts. And within individuals, it's well known that cells can conflict through presence of different genetic materials inside the organism, and also genes will compete to get to the next generation. So it's all levels of conflict. And surprisingly, actually, the concept that will decide who wins, who will pass on their genes, who will be on top, actually very similar according to the different levels of complexity. For example, I don't know if you are aware of this famous classic example of the social amoebas, Dictyostelium discoideum, sont amoebas that actually live in a solitary form in the environment, and when resources are lacking, they actually group together from a slug. The slug will go away from the environment, and then it will sporulate, so that there's a resistant form of the amoebas will be dispersed to hopefully a better environment for the cycle to start again. So what people have been, John Strassman principally have been studying is how genetic heterogeneity inside the slugs will influence the phenotype of the sporulating body. And what they found is that when the slug is clonal, that relatedness is high, actually, the sporulating body has got a longer stalk and less spores. When there is more than one clone inside the slug, the stalk will be shorter, and more of the cells actually will differentiate in spores, which means that collaboration is lesser. It seems that there is a conflict between cells of different genomes that makes that the cells cooperate late, so they will make a smaller stalk, and also more of them will differentiate in spores. So more of them actually wants to get to the next generation at the cost of other cells in the slug. So we have the similar systems in our complex animal societies, but it's a bit more difficult to study what is going on there. And mostly we lack genetic control of the composition of the control of the genetic composition of the groups. Something important, but which must be obvious to you, is that genetics is not what decides what's going on at the level of the group, at the level of the behavior. So social hierarchies, which are actually also very widespread in social animals, such as primates, but ants as well, and also in other social species, such as this fish, where complex behavioral interaction actually decides cooperation and the result of cooperation. So our model is the clonal radar hand, Ocera biroi, which is unique in ants because it's obligatorily parthenogenetics. So actually, workers will lay unfertilized eggs, which give rise to more workers. There are no hierarchies, so everybody in the social group is reproducing, and they are thus clonal. Most colonies actually are composed of identical individuals who have the same genome. And they are just an ideal model for studies of development in animal societies. They also have an army ant cycle, which means that in the colonies, there is a strict difference between the reproductive phase where eggs are laid and the workers actually are in the form of a pupae, and the foraging phase where the eggs hatch and the workers will go out of the colony to forage. We pass this. So this is what a colony of Oocyarea looks like. You see here all marked workers, a larvae, so this is the foraging phase, and the workers will go out to hunt for other ants to feed the larvae that will develop into more workers. So what we start doing, looking at the control social group which have the same genotypes, the same age, is our experience as the experience of the environment which will change behavior. So we did actually a pretty simple experiment. We had a nest chamber here where the ants are, and several times during the week we actually opened the nest for them to go out to forage, and we assigned the individuals to two experimental groups, one which will actually encounter prey during the foraging, its foraging effort, and another which will not encounter prey, so which will be unsuccessful. We did that for a whole foraging cycle, which is 15 days, and what we observed actually is that the workers which were reinforced with prey actually took less time to go out as we make them do more sessions, and the unsuccessful one actually would stop going out or would last a lot more before going out. And when we looked at their behavior a month later, when they were foraging again, we saw that there actually spatial segregation between the workers that were successful or unsuccessful during the first set of experiments. The workers which were successful actually were closer to the exit of the colony, and they were less in presence of the larvae. So what happens actually is that workers that were successful were going more outside, and they also are in a place in the colony which does not have larvae. So actually they are more successful at foraging, they stay more away from the brood. So workers which were unsuccessful actually come smaller to the, closer to the brood, which probably is another reinforcement for them to stay inside the colony to take care of the brood. So you have division of labor based so on the perception and the experience they have of the environment and the social group, which is something that actually had not been studied before or shown experimentally before, which shows a strong effect of the environment on the behavior, on the future behavior of workers. So I don't have much time to talk about this. Let's see. So what happens in this species is that actually there are two morphotypes. A morph which is more highly reproductive, which is this one that are more similar to queens in other species, and one which is less highly reproductive. In terms of number of ovarioles, so their fertility, these high individuals are actually potentially more fertile than the low ones. And what we did actually to study how this was regulated in the social group, so who reproduces in workers which are actually genetically identical. Let me remind you. We actually mixed different clones of the same colony to see how they would react to the presence of competition inside the social group. So in a previous experiment that we did, we showed that actually the fertility of the social group is regulated through the proportion of highly reproductive individuals, which means that when there is a lot of highly reproductive individuals in the group, in the next generation, the social group will produce less of these individuals. And on the contrary, if there's a small proportion of highly reproductive individuals, you will have in the next generation a higher proportion of the individuals. So there's a flexible adjustment of colony fertility which is proportion dependent on the proportion of fertile individual in the group. So what we did, we used the principle of the chimera. We mixed genomes of the same species to see what would happen. And we predicted that actually because of conflicts, the clonal line would actually try to overcome the other clonal line and produce more reproductive individuals. So this is the experiment. We actually mixed these two clonal lines, clonal line B, clonal line A, and we have control lines to see what would happen when we mix them up, to see how the social environment and potential conflict linked with genetic heterogeneity would actually influence how the group does and how each clonal line inside the group would do. So what happens is that we have here pure colonies of clone A, pure colonies of clone B, and our chimeras, which are a mix of the two. What's interesting is that one of the clonal line A produce a lot more individuals than the other one, and the mix. So it seems that actually conflict inside the colonies made the colony less fertile and in the colony. What happens in the chimeras is that clone line B actually outcompeted the other clonal line. So actually we showed a conflict which actually was resolved always in the same direction, with clonal line B winning on clonal line A. And what happens is that in our clonal line, in our chimeras, there is a lot more of clonal line B which develop into highly reproductive individuals. So what happens actually, and this is the message of this experiment, is that clonal lineage A has a low inhibition of development of its larvae, in highly reproductive individuals, and these individuals in return also have low probability of developing in highly reproductive individuals. On the contrary, clonal lineage B has a high inhibition of the development of larvae in highly reproductive individuals, and the larvae themselves have a high probability of developing in a highly reproductive individual, which means that actually there's a calibration in clonal line of the inhibition of workers on larvae and the probability of larvae to become highly reproductive individuals. When we switch that, ¿qué está acontecendo? Okay, tá bom. What happens is that when we put larvae of the clonal lineage B in the adults of clonal lineage A, inhibition is low here, and the larvae have high probability, so the clonal line B will produce many highly reproductive individuals and actually behave as a social parasite, exploring, I can't remember the word, taking advantage of the other genetic line, whereas clonal lineage A actually cannot develop in highly reproductive individuals when they are raised by adults of the clonal lineage B. So what happens is that colonies are calibrated for the group functioning. Also, they seem to be calibrated to certain environment, because this clonal lineage A is very fertile in constant environment, and this clonal lineage B is less fertile, but actually is more resistant to lack of resources because these highly reproductive individuals actually live for longer. So this is, to finish, I think it's a nice story that we showed that the genome of the individuals in the group you interact with actually will influence what you become during your development. And when we talk about ecology, we see also that in a species of clonal, which have clonal lineage, actually, different behavior, different reproductive phenotype can develop, who actually can lead to parasites and perhaps the evolution of new species when you mix actually this clonal lineage together. Well, the conclusion, the environment influence the development of alternative behavior in social species and probably other social interactions. Social context is of crucial importance. The interaction of genome present in different individuals influences the own individual gene expression and phenotype. So this complex gene-gene environment interaction can be a general factor, and more importantly is that the individual, as they experience the environment, will change their behavior, and in turn will change the way they feel about the environment. So their feedback loop constant about the experience of the individual, which comes from the environment, and the way it will change its behavior with regard to the environment it is confronted to. Okay, excuse me for going over time. A lot of people actually contributed with discussion, with work, and also it's a chimera because some of the photos here actually are not people from the lab. I'll let you guess which one they are. And I will thank you for your attention.
So, thank you very much for a very interesting symposium, and I think very relevant for evolutionary thinking and psychology. So, if there are some questions, you can ask. Oh, here, sorry.
Hi, I have a question for Patricia. You mentioned a relationship between food type and social hierarchy. Can you talk a little bit more?
Thank you, Mauro Júnior. And the features of food sources refer to a quality that we say that turns that type of food source monopolizable or usurpable. So it reflects the size, more or less the size. So if it has to be large enough for more than one individual share the same food source for a period of time that will make possible for another one to try to claim and to displace that individual from the source. So it's a type of source that generates direct competition. So usually we say that it's a type of clumped food source. So if it's too small, so I can eat this so quickly, nobody will steal it from me. If it's too large, all individuals can feed at the same time, so there is no reason for direct competition. So it's the size and the amount of food that make it possible to control, but also someone try to displace you to have the control of the food source. Did I explain? Thank you.
Other questions?
It's for Briseida. You mentioned in your talk that less behavior variety predicted more proficiency at nut cracking. Now, I thought it was interesting. How do you explain that?
You thought it was the opposite?
Interesting.
Interesting, right. So what the index of behavior variety was built considering the frequency of behavior they were doing at the nut cracking site, because we were filming what they were doing there. So they could take the nut, take the hammer, and also do something with the other guy, try to play. And they could do many things there. But what they should do to be a proficient nutcracker was take the nut, position the nut on the hammer, on the anvil, take the hammer and pound it, and that's it. So take, place, take, pound. The more proficient the monkey, the more focused on only this sequence of behavior they were. And the less proficient monkey, they did many other kinds of things. And so this behavioral variability was considering only the nut cracking task. So, and so when they were, they learned how to crack nuts, and they got to the nut cracking site for cracking, let's say. So that's what they did.
Some other question?
I'm wondering if you can, on the clonal ant species, if you could tell us a little bit more about the chromosomal structure in that species. Is it diploid? Is it haploid? Is it haplodiploid? And then also, how unique is this species? Are there closely related species that are also clonal? What explains the evolution of this clonal reproduction system? What were the ecological factors that led to the evolution of this type of reproductive system?
Right, and well, these social group where they are all females, they actually are diploids, and the parthenogenesis occurs through central fusion during meiosis, which is something studies which have been done to actually determine how clonality works, which seems to reduce actually homozygosity in the descendants. So it's one of the mechanisms that actually makes that the load of clonality is not that high, and also the fact that they are social. There is two aspects of these ants which actually may be an element to explain this reproductive system. The first is that they are distant, well, they are relative of the army ant. And army ants, they all have a syndrome where they have cyclic reproduction, where the eggs are all laid at one point, and the colony will enter in a statary phase. So the workers actually do not go out during this phase. And when the eggs hatches and larvae are present, they will actually go out every day to look for prey, which are other ant species. I don't know if you've already come across army ants in the neotropics or in Africa. Something that is really impressive. So the Sarapakis has got this syndrome at a lower level because colonies are less numerous. The explanation about the fact that clonality was selected is that they actually occur originally in parts where there's a high monsoon. That's right, monsoon. So actually, sexual reproduction, which needs a reproductive to go outside, have nuptial flights to meet, and then individual queens to found a new colony. This is actually very, the probability of this to succeed actually very little. So it probably, because of this, that some mutants which would reproduce parthenogenetically were selected in this species. And then you have the problem of what a species is, because so we have clonal lines. Actually, Daniel has discovered 4 different clonal lines, so actually they could have evolved separately, and so it's something that was really, there is some, these are the hypotheses, but we actually cannot really know. There is a lot of species, there is few cases of species events which actually also are clonal and which comes from the same geographic area originally. So it's something that could have been selected repeatedly because of the difficulty of sexual reproduction in these environments. Okay? Thank you.
For the second talk on the development of skills being higher in high predation monkey populations, I'm wondering if an alternative hypothesis is that if you're more likely to die young, you reproduce faster. And so I'm wondering if the skills that are developed are maybe more reproductive skills or more survival skills, if that's a good alternative hypothesis. Do you have thoughts on that?
It's a good hypothesis, but in fact, although they promote independency earlier, they don't reproduce faster. The interval between survival offspring, so it would be interbirth interval, but is higher in Una than in Boa Vista. So it's not exactly what we would expect. Thank you for the question.
Hi, so my question is about tool use in capuchins, and I was wondering if you recorded any data about reproductive behavior, maybe number of offspring produced or opportunities to mate, because it occurred to me when you were talking about how tool use is such a social event, I'm wondering whether, for example, males, those who, I guess, develop proficiency more quickly might be chosen more often for reproductive opportunities, because that might indicate intelligence or ability to adapt to new circumstances. And that, I guess, would maybe reveal something about the development of how tool use would evolve and perhaps be sexually selected in some ways. So I don't know if you have thoughts, if you have that data or you have thoughts on that.
We have data about the reproduction, and we have been following these monkeys for a long time. But we were not focusing on this, but we believe that the social, what we noticed is that the monkey is not paying attention in a particular monkey. It takes the opportunities for learning. So the young monkey or the naive monkey, he stays nearer the proficient monkey if he's tolerated. So and sometimes when they grow up, they are not tolerated anymore. So we have this thing, but we also have, for example, adults together in the nut-cracking time, let's say. And we have some scroungers who have the possibility to, or the opportunity to scrounge because they are tolerant. For example, when you have an adult male cracking nut, and then it comes another adult male for scrounging, he will not be able to do that. Okay, so he will be beaten. He will be attacked. but an adult female, perhaps she will be tolerated. But if she's in estrus, I don't know if you are familiar with capuchin monkeys in estrus. So it's very funny because the female, she chooses a male to follow, and it is usually the dominant male, but not always. And she persistently follows this male, and she's very sticky, and in these days, perhaps he will not let her to be around while he's cracking, because she is really sticky. And then so what else should I say?
No, just to add that it's like Briseida said, so dominance is much more important for a capuchin male to be selected, and we don't have data to show that nut cracking is related to dominance acquisition in males. So I think we have enough data now, because we were interested in pursuing these ideas. I think it has nothing to do with sexual selection, at least in capuchins.
Yeah, I'm just remember some of the best crackers, if they were the dominant, and they were not. And we have really some crackers who were not reproducing at all. So.
So, is there any other question, comment? If there is none, thank you again very much for the symposium. Thank you everyone for your attention. And now we will have a break, a lunch break actually, so we'll go to eat. And don't forget that we have a special prize in ECA, especially for you who are students here. It will be for, I think, 25 reals, which is a special prize for this Congress, so use it, which is good. And then we, what's the other important information? Then we will actually start at 2 o'clock in the afternoon with the official opening, and also with the keynote address. So don't forget to be here at 2 o'clock. And basically that's it. Thank you for your attention, and we will see you in the afternoon.